My name is Charles Cywin. I'm a program director at NIH. I'm going to hopefully we'll be able to tell you a bit up today about the funding programs for translational research in neuroscience space. It should be noted that it all starts with you. You bring in new targets, new small molecule with pharmacology, new ideas for biologics or devices, but and we may be able to help you at the NIH to move those projects forward, and hopefully you'll learn some things today. But it all basically begins with you, and we're here to help. Translation requires collaboration. A collaborative team includes experts in disease biology, experts in each aspect of drug discovery and development, clinicians who work with target patient populations, licensing and business development advisors. And you can consider partnership with the NIH for such things as with biotech and pharma, as well as through small business things like STTR and SBIR can combine all these things together. But as we said, the NIH will hopefully be able to help bridge some of these gaps and make these connections. And one key tool I like to always tell people about is NIH Reporter, because it's something you can do. It's available at your desktop. It's open public and has sort of public information in there. And you can put in simple things into the search field, something like allosteric modulator, Alzheimer's, and you'll get a bunch of grants that are available that are presently awarded, and information about those. And if you were to click on one of the names of those, you can drill down on that. So if you click on the link for this particular one, you get to a page that looks like this. And if you click the details tab on the left, it'll give you an abstract and tell you about the project and what they're doing. It also gives you both the PI's contact or PI's name and the company. But if you want to get more information, you can do similar projects and get further information about that. But if you click the details tab, as I said prior, you get this information, and this information includes you can get the email of that PI, but most people are also looking for NIH contacts. That I always see the question, I don't know who to contact NIH. By doing this kind of process, you can actually find out who's doing similar things to what you're looking for, and get a program official name and email that probably has that in their portfolio and can help you further your quest. I'm in the NINDS division, National Institute of Neurological Disorders and Stroke, and then in the division of Translational Research. That's my home institute. I'm going to talk to you today about both DTR projects as well as projects that are trans NIH, meaning their management is through NINDS, but they actually take projects from multiple different ICs and are funded across different ICs in different ways. So we have a whole suite of projects that take you to discovery all the way through the clinical trials, as well as some services in terms of the epilepsy screening, epilepsy therapy screening program, and the preclinical screening platform for pain. And I'll talk about all of these programs today and give you a sort of where they fit into the puzzle. So the first program I really want to tell you about is an early stage program. It's called Ignite, Innovation Grants to Nurture Initial Translational Efforts. It's a suite of funding opportunities that's designed to get you the preliminary data necessary to come into the later stage translational programs like Blueprint Neurotherapeutics Network or the HEAL translational programs. So we'll look at these individually, but they're all designed to get you that robust preliminary data to make for a good application. There are three different applications here. There is the assay development and neurotherapeutic agent development. So think of that one like develop a screen or develop an assay and run a screen. That can be done within this. We have the neurotherapeutic agent characterization and in vivo efficacy study. So think about that one sort of like, I need to do some additional optimization to get good in vivo tool compound to run a in vivo efficacy study. So I have robust preliminary data for my application. They also have a PIR for developing an animal model. So if you don't have the model in your lab and you need to develop this model, this is an opportunity to do so. All of these grants are what are called phase mechanisms, R61, R33, meaning there's a milestone between the two phases. And so you have to have a clear go, no-go milestone. You have to delineate that in your application. These are maximum of $750,000 for the entire project, maximum of $500,000 in any given year, but you can split that up. The next application due dates are in February and June, and Rebecca Roof is the contact at NINDS, the program director, that will help you with that project. The one that's near and dear to my heart, the one that brought me to NIH in the first place, is the Blueprint Neurotherapeutics Network, otherwise known as BPN. It combines the strengths of NIH and industry expertise for small molecule neuroscience drug discovery. It starts with NIH investigator-initiated ideas, a solid scientific premise, and disease expertise. From the biology side, you must have the assays and models as part of the grant. And what we do is really unique about the program at the time, and still is somewhat today at NIH, is we bring together expertise from outside NIH. We have consultants with extensive pharma experience in various disciplines like medchem and DMPK and toxicology, et cetera. And we have a pre-established industry standard CRO network that we've put in place that we can give you access to as well for, again, doing all the different disease agnostic type activities in order to get you to the clinic. The end goal of the project, again, is to keep the intellectual property with you. And to decrease the risk as the project advances and advance the project to the point where we get to the clinic and hopefully a handoff so that someone's going to take it farther and on to phase 2 and 3 and move forward. So how do we do that? Well, we established what's called a lead discovery or lead development team. And we meet every 2 weeks. It's just like a pharma project team. All the people we talked about are in there, the consultants, your staff, as well as NIH staff. And the NIH staff are there as facilitators. We are, there's usually a project lead or project manager. They are the person, your single point of contact you can come to about things. But there's also science officers, people I mentioned in that earlier slide in the reporter that have the portfolio of your disease area, that really know what's going on in the area and can give great insight on, is this up to sort of what is the state of the art? Are there things we should be considering we're not thinking of? They have that perspective. It's a custom tailored approach. So you have to have as I mentioned, the bioactivity, the disease biology, the primary SAR driving assays from a biology point of view, the in vivo efficacy studies have to be part of your grant application and your grant budget. But things you might not have, like medicinal chemistry resources or PK and tox, drug manufacturing formulation, eventually clinical trials, those are contracts we have in place we can give you access to. If you're doing discovery effort, we also have a relational database that allows you to put the information that the medicinal chemists are generating compounds and the data that you're generating, along with the in vitro ADME data that we generate, all into one place. It should be noted that the medicinal chemistry contract we have here is not just synthesis, it's also in vitro ADME, it's also computational chemistry. The PK and tox is rodent, large animal, for both the pharmacokinetics as well as the toxicology. We do dose ranging and GLP toxicology to enable phase 1. And things beyond phase 1 will be out of scope for us. So the question you really have to ask is, do you have the expertise and resources to do these activities in-house, or do you really want to leverage? In most cases, I always tell people, try to leverage our resources the maximum you can. You don't want to hire, for instance, medicinal chemists to do the work, because if you miss your milestones, you'll end up having to get rid of those chemists, whereas we have a standing relationship and can go up and down very easily. As I mentioned, this is one of the trends in NIH programs. There are 9 participating ICs listed here with their contacts, which should be noted is they all have interest in doing translational work. And we have small business as well as non-small business funding opportunity announcements here. And so there are contacts for either. So what we do here is we have a U44 fast track mechanism, and we have a UG3 UH3 mechanism. This is U44 is only for SBIR eligible organizations. And you have to follow all the SBIR rules. And what you see here are the budgets for NINDS, but other ICs have different budgets. So if you're going to come in through the SBIR, please talk to me first, and we can give you some guidance. It's also important to notice that not all ICs accept development projects. So NIMH and NIDA particularly are not going to take a development project that comes in just down here. But projects, generally speaking, can come in anywhere along the pipeline for most ICs. Small businesses who are not eligible for SBIR are still eligible for the UH3 mechanism. And there are advantages to both depending on your situation. You know, the SBIR, you can charge a fee. If you can meet all the SBIR rules, we recommend you come in that way. But sometimes because of ownership, sometimes because of the budget, the way it's going to lay out, and you won't be able to meet all the rules, you may need to come in the UG3, UH3, and that is okay. Again, we have receipt dates coming up in February and August. These happen twice a year for the Blueprint programs. And again, it's a milestone-driven program. This, as I mentioned, is a phase mechanism. Typically, the first phase is a year for the UG3, and up to 4 years for the UH3. There are milestones for each step of the way. If you come in and say, well, I'm already a hit to lead, we're going to be looking to see that, you know, you already have assays in place. You can demonstrate good statistics on those and good throughput, that you have your screening funnel and target product profile in place, and that you've demonstrated at least a preliminary SAR to have an SAR plan to come in to hit to lead. Once you're in hit to lead, we're really looking for pharmacology, starting to see efficacy in vivo by any route of administration, and trying to get yourself from multiple series down to the 1 or 2 series you're going to use in lead optimization. And it progresses like that all the way through. As you get to the development phase, it's really all dictated by the FDA requirements to get to an IND. And as I mentioned, we do have a clinical trial contract that you can utilize. These grants are not considered clinical trials from the grant side if you are doing the clinical trial through our contract. So it's important to note there are differences when you're doing the application. Things that you're saying NIH is going to do, you don't have to go into great detail about. Things that you're saying you do need to adequately justify that you have a good plan for those. Likewise for the human subject. If you're not using, if you're using our contract, this is not going to be human subject application. It's not going to be a clinical trial application unless you have human subject somewhere else in your biological screening funnel. I mentioned intellectual property. Intellectual property is, you know, inventorship is always determined by U.S. patent law. We are not seeking a stake outright, and so prior to award, we're going to look that your institution has upfront IP agreements with all players involved in the project, including the ones that you're putting on your grant application. So if you have multiple institutes, then we're going to look to see that you've already got an agreement in place amongst yourselves. And, you know, our contractors, the consultants, they'll sign IP agreements with you to say they'll sign it. You know, we don't get involved in those, and they may or may not require, you know, royalty arrangements. That's really up to you. How that works. You're not paying them. Their confidentiality is part of their contract within NIH. Our MedChem and drug manufacturing formulation contract, with places where IP is really being generated, have special clauses associated with them called determination of exceptional circumstances that allows them to assign intellectual property to your institution directly out of the project. So the overall goal is to have unencumbered IP. Blueprint staff are really going to be facilitators. We're not going to get involved, even though, as I mentioned, I have medchem experience. I'm never going to be telling you to make this compound. That would potentially make me an inventor. I will, in general sense, work through your consultants. In a general sense, may ask, you know, are you considering different strategies, but I'm not going to get into the weeds of you should make this compound or that compound. Our website is here. If you go to the Blueprint for Neuroscience Research, you'll find you land on a page like this. This is the Blueprint, the small molecule one, but if you click up here, you can get to the biologics and the medtech version too. All the resources that I talk about, BPN resources, our external oversight committee. You can find out information about all that, including all of our contacts. As I mentioned, BPN was designed for small molecules, but last year we've expanded it now for biologics, and so we have a new program that just launched. This first receipt date was back in August. It's set up very similarly, and we're putting the contracts in place right now. They're utilizing some of the small molecule contracts, and additional contracts are going in place. The biggest difference is the UG3 phase can be up to 2 years, and that doesn't happen in the small molecule space. Our contact here is Mario Skiadopoulos. His email's here. As I mentioned, again, lead discovery, lead development teams are set up. The contracts are in place. Same idea for intellectual property. The application dates are identical. The PAR numbers are PAR 21-163 and 21-162. We also have a large suite of device programs, and I'm not an expert in device, and so if you have questions about devices, I would highly recommend you reach out to NINDS device group at this email. But there's a whole suite from concept generation all the way to clinical trials. We have programs through the Brain Initiative that are dealing mainly with, I think, invasive devices. We have the translational neural device program where the reissue has been approved. It may actually even be on the street at this point. So there's a great set of funding opportunities here to move your projects forward in the device space as well. The SBIR STTR program is a program where a lot of people come into. You've seen a couple of targeted FOAs with the BPN that have SBIR eligible funding opportunities, but there's a whole program in the omnibus, which takes basically all comers, is part of that, and it's a congressionally mandated program, the SBIR STTR program. It has a set aside, meaning there's funds have to go to these small business grant opportunities, and we fund a lot of projects through there in the translational space, and they have broad scope. They can take therapeutics, diagnostic research tools, bench research, early stage clinical trials. So it is a huge opportunity and one I recommend you consider if you're a small business. It does allow for some larger budgets on some topics like animal studies and clinical studies, as you saw for the BPN, guidance for NINDS. I often get questions about STTR. Again, small business is always grant is always made to the small business, but there are different rules when you're doing STTR, working with an academic organization on what the percentages have to come from each side, where the PI has to be employed, and such. So I do recommend you reach out to the small business representative for the institute that your disease area is in and discuss your program with them. We have a program that's housed in NINDS, which is NIH Countermeasures Against Chemical Threats. This is a interagency project. It goes across the government, and this aspect of it here is we're trying to deal with fundamental mechanisms of toxicity caused by chemical threat agents and try to develop promising therapeutics to reduce the mortality and morbidity caused by those agents. There is a R21 mechanism with the next receipt date in May. There are core facilities that are available at no cost to the investigators. And again, we're trying to retain intellectual property with you. Dave Jett is the program director there. If you have questions about this program, I recommend you visit their website and contact Dave. I also mentioned you have two additional screening programs. The first program is the Epilepsy Therapy Screening Program. This has been in existence since 1975. It evaluates novel compounds in preclinical models to test their potential as a treatment for epilepsy. It's, again, at no cost to you. You can submit them. It's done through an NINDS contract with the University of Utah, and Karen Wilcox is a great expert in this area as the PI, and runs that facility. We are now focused on drug refractory epilepsy and disease prevention. They've tested over 32,000 compounds, and they've had a role in 9 marketed drugs for seizures. Additionally, we have the preclinical screening platform for pain here, and as part of the overall portfolio of HEAL, I'm going to talk to you about a few different things here. I'm going to give you a highlight, but the first one I want to talk to you about today is the PSPP. Again, it's a one-stop resource for clinical screening of potential pain therapeutics. It's now accepting agents for evaluation on an ongoing basis. It's no cost. The intellectual property is, again, protected. It can go through and have side effect profiles, abuse liability, PK, animal endpoints, opioid activity, amongst others. Smriti Ingar is the contact here, and please reach out to her. The HEAL, the Helping to End Addiction Long-Term program, has in it everything from early stage through the clinic. I'm going to talk today about the preclinical area here. We have a number of different grant mechanisms. We have planning grants to get ready to team building, to think about mechanisms of feasibility. We have the U19 program, which is a large multi-group kind of approach to bringing together people to have the entire package, to deal with mechanisms, assays, models, biomarkers, proof of efficacy, all in one big application, and get you ready to do to an optimization program. I'll focus in on this one because this is much like BPN. It was modeled after BPN, and I have responsibility for this one. Again, the same result is in all of these preclinical things, is to take you through the process and get you to the clinic, and hopefully get you licensed or move you into phase 2, either through EpicNet or through that licensing process. The biggest difference between this and BPN is A, it's only focused on pain. B, it has some different, it will take any pain indication from any IC and NIH. So it's not just the ones that are participating. So we'll talk about that in a second as well. It is milestone driven. It also has a desire to find and identify a companion target engagement biomarker for the program. We're utilizing the same contract structure as BPN and leveraging, adding additional contractors to the different disciplines as we can. It takes small molecules and biologics, so therefore we're in this, as we add the contracts for BPN Bio this spring, they'll be able to be leveraged in this program as well. We hope to launch up to 17 projects, and we just started in 2021, and we hope to advance at least 5 of those to the UH3 phase onto IND in the clinic. We have receipt dates coming up 3 times a year in this program. February 15th, June 15th, and October 14th are the next 3 receipt dates for this program. Much like BPN, you can enter anywhere along the drug discovery and development paradigm. And what is different is you will see there's no U44 here. All applicants, small businesses, non-academics, all come through the UH3 mechanism. It does have a 2-year max UG3 rather than a 1-year max. And typically UH3 is 3 years. Applications must have both halves. You can't come into UH3 and you can't just do a UG3. Foreign institutions and components are allowed. Do talk to us before you apply so we can tell you about the additional requirements that you'll need to demonstrate, and so you can cover that in your application. All projects are expected to lay out a path to the clinic via the grant or the NIH contracts. Discuss the budget with us. they're not limited in this particular thing for pain, but we want to make sure we understand exactly what you're proposing. There are 9 participating ICs, as I mentioned. They're all listed here. What that means is these institutes have said, and these folks you see here are willing to come in and work with us on the project teams and give us science, their science expertise. But if they're not available, NINDS staff will fill in for that and be the science officer for the pain indication. I don't want to go any longer. I do want to make sure that I mentioned that the NIH has a number of other drug development, drug discovery FOAs around NIH, and I encourage you to look at not only IC but other ICs out there like NIDA and NIAAA and NIA all have funding opportunities. I'm happy to answer any questions about the application, the review process, the proposals and contracts, and anything around intellectual property you want to know. Thank you, and I'm sorry I went a little longer than I expected.